Hello, I'm Richa, and Gary and I are clinical scientists, and we work in the neurogenetics laboratory here in Queen Square. We do the genetic testing for the skeletal muscle channelopathies, and I'm going to discuss what this involves. So the ion channel diseases we work on include Project Smiths, and I've listed the genes responsible in brackets. Anderson-Tawil syndrome, caused by variants in KCNJ2, paramyotonia congenita, which is associated with the ESM4A gene, and myotonia congenita, caused by alterations in the chloride channel CLCN1 gene. So when the patient comes to clinic, a blood sample is taken for genetic testing and sent to our lab. Our neurogenetics and the Great Ormond Street Hospital lab team are based in the same building. The technical team do the practical laboratory work, and we as scientists analyse the data and report the results. The DNA is extracted from the blood sample, and then the technical team carry out a procedure called DNA sequencing in the lab on the extracted DNA. All the channel genes are tested for at the same time. The data is then made available to us, and we check the channel genes to see if there is a change in the DNA that causes muscle channel disease. So here is an example of DNA sequencing, and we call this technique next generation sequencing. The image shows a section of DNA from the channel gene. There is a sequence of letters at the top of the image. Each letter represents a part of the DNA called a DNA base. So the base or letter highlighted by the arrow is a G. So an unaffected individual would have a G at this position. A DNA change from G to A has been detected in the patient in a channel gene, and we call such a change a DNA variant. So once we have detected a change in the DNA, we make an assessment to check if it could be disease-causing or not. The assessment involves using a number of resources, databases, and online tools. For example, scientific literature searches, and review for gene variants associated with channel disease. Checking our own lab data, checking if the DNA change is common in the general population or not, and using online DNA analysis computational tools. Once the decision has been made that a variant in the channel gene is disease-causing, we will write a report to the referring clinician, and this will contain relevant information on the gene change that we have identified. So this analysis and reporting is carried out by one scientist and checked by a second scientist. Then the doctor will give the patient their results in their clinic appointment. So I just wanted to show you another example of DNA sequencing. So sometimes we carry out testing for the gene change in family members, and this is an example. This technique is called Sanger sequencing, and we just test for the DNA change in a family rather than test all the channel genes. So the DNA base or letter highlighted by the arrow is a G in the top image. So this is the DNA of an unaffected individual. The G base is represented by the black peak. The lower image shows the patient sample with a DNA change detected in the channel gene, and this is represented by the A letter, which is the green peak. So the patient has inherited the green peak, or A, from the affected parent and the black peak, or the G letter, from the unaffected parent. That's a summary of what we do in the lab. Thank you.